Hi, this is my presentation on the gene FAM83A, and it is an essential gene. It is conserved and slow evolving. In this lab report, I applied my biology course acquired knowledge of bioinformatics skills in order to analyze a gene. For context, genes are sequences of nucleotides in DNA which code for a functional product. Humans have an estimated trillions of genes, yet only a small percentage of them are encoding. These genes that encode for a functional product are deemed as essential. Now, to determine whether a gene is essential or non-essential, there are some techniques that one could use, such as the knockout method, where you can essentially engineer the gene in question, removing it from the genome and observing whether it causes harm or if it has a neutral effect on the phenotype. In this report, however, we dive into the statistical analysis of things and build a molecular clock. So let's really talk about it. Alzheimer's disease, or AD, is the cause of 60 to 70% of diagnosed dementia worldwide. Its common early symptoms are difficulty remembering recent events, which most of us are familiar with. However, as AD worsens, it can cause for the patient to have mood swings, disorientation, and they get so disoriented to the point where they will forget what they did a few minutes ago. I actually chose this phenotype because I know a close family member that suffers from this mysterious disease. He will eat and forget minutes later that he just ate and announce to everyone that he's hungry because he has not eaten. In theory, it's a little comical to hear such story, but actually experiencing it before you, in front of you, is pretty devastating. Such situation can easily turn from overeating into severe stomach issues. Other symptoms of AD include behavioral issues, aphasia, which is difficulty with language or speech, self-neglect, loss of bodily functions like weakened muscles, and overall health decline. According to Alzheimer's Disease International, as of 2020, there are over 55 million people worldwide living with dementia in 2020. This number will almost double every 20 years, reaching 78 million in 2030 and 139 million in 2050. Much of the increase will be in developing countries. For that phenotype, my gene of interest is FAM83A. It is located in the UTR5 gene. It's actually untranslated. It's on chromosome 8, band 8q24.13, so it's on the Q arm. This gene belongs to a superfamily, and superfamilies are genes that typically contain several protein families, which shows sequence similarity within each family. My gene of choice belongs to the DUF or DUF and PLDC superfamily. According to PubMed literature and my research on it, it only has 3 articles written about it, and there are 634 citations about my gene of choice. FAM38A has 37 SNP associations. The picture on the left is simply just a screenshot of the location of it that I accessed from Wikipedia. Now let's move on to SNPs. Genome-wide association studies identifies individual SNPs that account for a small percentage of risk to diseases. Large numbers of SNPs across the genome can help us know the overall risk of disease developing. Essential genes having certain variants or SNPs of that gene can alter function and structure of proteins, which have been linked to diseases. So if a gene is used to being conserved and then a mutation happens, there can be some very harmful effects that it could have towards that phenotype. So the whole purpose of this lab report is to determine whether my gene of choice, FAM83A, is essential after all. By using evolutionary context and causal versus correlation analysis, I'm going to determine whether the SNPs of my gene has a causal relationship with Alzheimer's disease, my phenotype. With my hypothesis being that my gene FAM83A is an essential gene because it is located in the UTR5, where single nucleotide polymorphisms, or SNPs, are associated with higher risks of diseases. The methods I used are these. So for sequence alignment, I used 14 homologous protein sequences, which includes the human gene sequence for FAM83A, my gene of choice. Nucleotides were matched up to determine similarities and homology within the 14 taxa. The image to the left contains my 14 taxas, their accession numbers, and the lengths. Homology implies conservation of sequences or genes, and it also means that the 14 taxa all share the common ancestor, as well as have similar functions in their proteins. For this, I downloaded Eugene and used procedures provided in the Chaminade Biology 308 lab manual. I used default settings and cluster W sequence alignment using human as my reference sequence. After sequence alignment, on the far right tab on Eugene, it allows you to generate phylogenetic trees. For the tree settings, I used default and the Jones-Taylor-Thornton setting. I did an IQ tree, neighbor joining tree, and the Mr. Bayes tree. From that, I could export my Newick file. After building my trees using Eugene, I used comp-phi, or phi, which allows for comparison between the trees against the consensus tree, which we were provided in class. I chose the tree with the lowest score as my best one, and I uploaded that Newick file for that best tree into dist parser, which generated all the branches and distances in a concise manner for me. I got 91 results because I had 14 taxa. Lastly, I did a rate test where an outlier group was compared with 3 taxa and human taxa. So I had 4 total in groups, the lizard being my outgroup. This is to see if there is correlation or causation happening within the evolution of these species. The null hypothesis will be that the difference between 2 taxa since last common ancestor is the same, and there will be a constant rate of change in the sequences. Different species derive from each other through speciation, which is the isolation between one interbreeding population to 2 separate populations. So there's no gene exchange because they're isolated. So there's not going to be any inherited shared genes over generations, which leads to species looking very different from each other. The purpose of this rate test is it will distinguish between a species evolving randomly or neutrally over time compared to a non-random process. These are my alignment results using human as my reference sequence. And this is from Eugene. These are my gene tree results. They're all in Newick text format. And these are my gene tree reconciliation results. My best tree is the Mr. Bayes, as it scored the lowest. This is my gene tree. And looking at the gene tree, you can see why I used lizard as my outlier. After some calculations, I ended up getting some pretty high p-values, way higher than 5%. This means that the difference between two taxa since last common ancestor is the same. As you can see from my graph for my molecular clock, you can barely tell apart the trend line between the line coming from the origin and the line of best fit for the distances. From our class results, my gene is on the slower side, which is a good sign. This is a telltale that my gene is potentially essential or is essential. The slower the gene evolves, the more conserved it is, and essential genes are conserved. My GOI is actually right in the middle of our class results. In conclusion, my gene of choice, which is the FAM83A, is an essential gene according to our statistical analysis. It is slow evolving, which implies conservation of proteins over time, and that the gene has gone through neutral mutations over time. The high p-value also means that the difference is not statistically significant, and we can safely assume the validity of the molecular clock. Lastly, scientific literatures have found that FAM83A is involved in cell population proliferation and epidermal growth factor receptor signaling pathway and is predicted to be located in the cytoplasm. Numerous article citations concerning this gene has linked it to diseases outside of AD like breast, pancreatic, and ovarian cancer. Since the gene is essential and yet there are only 3 articles on PubMed about it, it deserves further research. Thank you.